Dear students, in this video, I am discussing about a topic useful for botany, microbiology and biotechnology students. Topic is diatoms. If you find the information is useful, please subscribe the channel and like the video. Diatoms are found in salt water and fresh water. Some are planktonic, that is free floating. Others are submerged forms, benthic. Some occur in soil. Many planktonic species are sensitive to light and salinity of water. Diatoms are the chief source of food for marine fishes. Diatoms form fossil deposits due to the accumulation of siliceous cell walls. These deposits are known as diatomaceous earth. Structure. Most of the diatoms are unicellular and varying in shape. The diatom cell is like a box having two halves. The upper half overlapping the bottom half. The whole diatom cell is also referred to as frustule. The upper half is epitheca and it fits closely over the bottom half called the hypotheca. In pinnales, the diatom cell is like a oblong box. In centrales, cell is like a petri dish. The edges of epitheca and hypotheca of diatom cell overlap forming a girdle. The edges are joined to connect band or cingulum. The centrales are more abundant in marine water and are radially symmetrical. The pinnales are freshwater forms and are bilaterally symmetrical along the long axis and in transverse plane at right angles. Diatom frustules have silicified walls and growth of diatoms is dependent on silicon concentration. The process of formation of silicified wall is controlled by cytoplasm and requires energy. Next is the types of markings on the valves. First one is punctae, small perforations of the valve surface and forms fine lines called striae. Second one is areolae, larger depressed structures. Third one is costae, heavy deposition of silica. Fourth one is canaliculi, narrow tubular canals passing through the wall. In this slide, you can see the cell structure of diatom. Protoplast of diatoms. It forms a lining layer inner to the cell wall. There is a conspicuous central vacuole. In centrales, the nucleus lies in the cytoplasm inner to the cell wall. There are several discoid chromatophores. In pinnales, a broad band of cytoplasm cuts transversely across the vacuole and the nucleus is embedded in it. Chromatophore are large lobed plates and one or two in numbers. Mitochondria and Golgi bodies are reported. Next is pigmentation. Chromatophores are golden brown in color. Fucoxanthin and dinoxanthin give this color. In few, green color is reported due to the presence of chlorophyll A and C masked by the xanthophylls. Some species are colorless and they are saprophytic. Assimilatory products in diatoms. Droplets of fatty oils, volutin, leucosin are present. Cell division. The nucleus divides first followed by chromatophores and pyrenoids. This is followed by the longitudinal division of the protoplast parallel to the valves. Two daughter protoplasts separate and one of them gets the epitheca and the other hypotheca. Oxospores. When diatom cells become very small, they resort to sexual reproduction which leads to the formation of oxospores. They are without valves. They can enlarge and form new valves. The original size of the diatom is restored. Microspores. Members of centrales produce microspores. These microspores are uniflagellated or biflagellated. Resting stage. Marine centrales produce endospores or statospores and produced by the contraction of protoplast and secretion of a thick wall. Economic importance. Useful as food to several fishes and other aquatic animals. Diatomaceous earth is used in sugar refineries for filtration of liquids, added in paints to increase the visibility during night, used in preparing toothpaste and silver polishes.